The testing context has moved on massively in the last 5 to 10 years. Lots of different kits, different manufacturers. There really is a need for a appropriate way for laboratories to check the quality of their testing process, because of the differentiation between the labs. I think there are two components of it. The first of all is ascertaining whether a laboratory, if it's testing a particular gene, is picking up the same variants of any other lab testing the same gene. And the second component is looking at pure data quality. And that data quality is things like the VCF files, the BAM files, FASTQ files, the raw data, and ascertaining whether their data quality is of a similar structure or similar quality to the other laboratories doing that sort of test. Those are the two main key areas I think that are important. I think using controls is really valuable, really valuable. There's definitely been not enough emphasis placed in the past on being able to use proper controls for checking and validating your assays. I mean, validation of your assays is the key thing for many of these laboratories. But if they're buying an off-the-shelf product, they need to be at least minimally verifying the tests. And these types of calibration controls are crucially important in the process of checking the laboratory's quality. There are still no real guidelines for diagnostic practice for next-generation sequencing. There are American guidelines, and there are some European guidelines, but there's no harmonised approach to that testing process. And I also think the key driver for that also is the fact there are no harmonised data standards. So we all use in our bioinformatics processes VCF files or BAM files or FASTQ files. And whilst there is, for example, a standard for VCF, every manufacturer uses a different version of that standard. And until we have standardization of those data standards, it'll be very hard to do any standardization of the whole testing process. I think the industry is moving towards a more integrated approach for the testing process. So next generation sequencing is a big thing at the moment and has been for the last 2 or 3 years. But I think it's just another tool in your box of tools that you can use for doing the testing process. And I think where we will be looking at in 5 or 10 years is a more integrated approach. We'll be seeing more different types of testing involved in the patient pathway, all brought together to answer that clinical question. So more use of liquid biopsy, ctDNA testing, more use of NGS for answering some of those difficult questions. But all the traditional testing still won't go away. It just has a different context. So I think we're going to see a broader range of technologies used in the testing process in the future.